This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 490, recorded on April 18th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, where it is 10 degrees Celsius and lovely blue cloudy day, Dickson Despommier.
Hello, Vincent.
It didn't make any sense.
No, it doesn't. But what was
blue
really
sky with some clouds.
It was a special day today because as we were working on my soon-to-be resurrected Racaniello page website, I glanced out the window and observed a bald eagle go by.
Yeah, it's pretty cool.
It's very cool.
Now, I thought that you can't say resurrect for a web page because it's not living to begin with, right?
Yeah, well,
neither the word is used for other things. I know, the word is used for other things. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's pretty nice here, too, although it's still quite cold. It's 38 or 36 Fahrenheit, and I can't get the app to do the conversion right off the bat. Yeah. And we have the same thing, light blue sky with puffy clouds.
Right.
Yesterday, I did an immune, and Cindy Leifer in Ithaca, it was snowing.
That's right.
And she said it was sticking. Can you imagine midway through April?
we had snow on Monday, Tuesday, and I think yesterday morning as well.
Eek.
Oh, yeah, yesterday was Tuesday, yeah. And a bad ice on Sunday morning, quarter-inch ice on sidewalks, everything.
That's not good.
Yeah.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Welcome back, Rich.
Y'all.
Thank you. Thank you. I've missed you all. Y'all.
Well,
y'all's
missed
you
too.
It is 76 Fahrenheit, 24 C here, and it is high overcast.
Any wind?
It's been generally a nice week. That's a really good question, Dickson, because it turns out that Austin is a pretty windy place.
Yeah, I knew
today
that.
Today there's not a lot of wind.
Right.
Yesterday and the day before, it was blowing like stink or smell, not necessarily stink. It was. I saw one of these little sort of jokes the other day. That was a Lubbock, Texas windsock that consists of a pole with a big chain hanging from it. Okay. And then it had all sorts of stuff about what the weather was like in the relative to the angle of the chain. Like if it was 0 degrees, then the windsock was broken and you ought to call the meteorologist. Okay. And if it's 85 degrees, it's welcome to Lubbock,
In 10 episodes, we'll be in Austin.
That's right.
We will.
10 episodes. It'll be
10 weeks, right?
Warm here, I
10
think.
weeks.
That's fine. I love it.
Yeah.
You Lubbock or you love it?
I love warm weather. I do. I really do. I'm ready for.
I can tell you, I've never had wind as bad as it was down in Argentina. The southern part of Argentina, the wind was consistently 40 miles an hour.
That's a lot. And
sometimes up to 60 miles an hour. So they advise you to park your car looking into the wind.
Wow.
If you opened your door the other way, it's liable to blow it off.
Yeah, break.
And they've had that happen.
I guess there are not a lot of trees there.
None. It's the pampas. It's absolutely grassy plains. That's it.
ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, June 7 to 11 in Atlanta using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial sciences in the world. Delve into your scientific niche in 8 different tracks. I hope you don't drown in your niche.
I would. You can't do that, actually. That's not the definition of a niche.
Don't miss this opportunity. Visit asm.org/microbe. That's asm.org/microbe and use promo code ASMPOD for $50 off registration. See you in Atlanta. Rich and Kathy and I will be there.
Looking forward to that.
On the weekend for TWiV.
Yeah, it'll be fun.
Happening with ASV, Kathy.
Well, I wanted to tell you a little bit about the ASV job site, which has recently been revamped. So we've had the job site for a long time, but we needed to modernize it. And so it's up and available now. It's much different from the past. I think it's more modern and user-friendly. It's currently free for everybody to find jobs. We intend to keep that way. And it's currently free for everyone to post jobs. In the future, we may have only free posting for ASV members, but currently it's free for everyone. And if you had an ad posted on the previous job site, we contacted you and you had a chance to migrate it. If you didn't do that, you should just do that now. This is a pretty cool site because I went on it today. You can filter by various criteria. We currently have some international jobs posted, for instance, one in Marseille, France, and one in Seoul, South Korea. A lot of varieties of categories, not just postdocs, not just faculty positions. So check it out. You can do that by going to the main ASV site, asv.org, and click on the Jobs tab, or jobs.asv.org. Either one of those will work.
The microbiology department at the Icahn School of Medicine is seeking faculty candidates interested in strengthening their program in virus-host interactions. Applicants must hold a PhD and or MD, have postdoctoral experience, and be interested in creating a virus-related research group that will complement the pre-existing departmental program. Recruitment of individuals or couples at the level of assistant, associate, or full professor will be considered. More information about the department and the school can be found at icahn.mssm.edu. If you want to apply, please send your CV and a brief description of your research plan to nycvirology@gmail.com, or you could contact Ben Tenuver, who is running the search.
Vince, isn't that where you got your PhD?
It is. At the time, it was called Mount Sinai.
Exactly.
School of Medicine.
Exactly.
And back when I went
back
to
in
school,
the day,
you didn't sell names.
No.
It was it was not the PNC Arts Center. It was the New Jersey Arts Center.
Exactly.
All the stadia had nice names.
It's true.
And even this venerable institution of which we are a part has sold 2 names in recent years.
Yes.
We are now the Columbia Vagelos College of Physicians and Surgeons.
And the Milstein Medical Center.
And the Columbia University, what's the guy's name? Milstein Medical Center.
Yep.
Dickson, we can put your name on something.
Could. It would put my name on like a postage stamp or something. Not even on the back of the postage
Speaking
stamp.
of money.
Yes.
The Viruses and Cells Gordon Research Conference needs your financial support. NIH and foundation support has declined dramatically in recent years. The organizers of the 2019 meeting, Julie Pfeiffer, chair, and Britt Glausinger, vice chair, need $70,000 to support partial registration and travel costs for 32 speakers, 18 discussion leaders, and selected students and postdocs. Donating takes less than a minute. Go to the secure donation site, which we will post on our website because it's rather long, and provide contact information, the amount of your donation, and how it can be used. Your donations are tax deductible, and you will be acknowledged as a friend of TWiV on the conference materials. I should make a small URL for that so I could mention it, because a lot of people drive when they listen and they can't, and they'll forget about going to the site by the time they get to their destination.
I find that rather a disturbing
trend.
Yes. The notion that the Gordon Research Conference should have to, have its handout.
Yep.
Rich,
that meeting, traditionally that was years ago, that was the premier molecular biology meeting. But although molecular biology has moved on to many different venues, that meeting has lost none of its clout. And it's always just a really amazing meeting to learn about viruses and cells.
Yeah,
and
great meeting. Great meeting.
Should be supported.
When I was involved in my own research, I became the vice chair and then finally the chair of our Gordon Conference for Parasitism. And the job of the vice chair was to write Dunning letters. You wrote these letters to industry and you tried to collect as much money as you could.
Straight up.
And that, well, I don't know about parasites. Maybe it has.
Yeah, this is viruses. It may be different for parasites.
It might. But I had no problems.
Because you guys have a nucleus and we don't.
You see? When you got a little bit more DNA behind you.
Well, it's just compartmentalized.
Yeah, this is true.
Anyway, do donate if you can. As little as $10, if you have enough people, we can get this money.
Yeah.
I'm helping them out to do this because Julie had sent out an email earlier saying, how can we raise money? We're having trouble. I said, try crowdsourcing.
Yeah, that's right. That's a good idea.
So I think it's good. Now, on a sadder note, we have a loss of one of our colleagues, Richard Courtney. He was a virologist, Penn State Hershey, right? Yes, Hershey.
Right.
Too bad. I'm going to go to Hershey this fall. He passed away on April 11th. He was a herpes virologist, right?
Yep.
Am I correct? Does anyone know anything about him? I know he was on study section with me for a number of years. I got to know him there.
I interacted with him several times, so I was acquainted with Dick. He was an outstanding scientist and a real mentor to both students and faculty as chair at both LSU and ultimately at Hershey, and a real gentleman, absolute gentleman.
So he was born in Pennsylvania. He went to Syracuse University. He got his PhD there, and then he moved to Houston. He was at Baylor for a number of years, where he went through to associate professor, and then moved to Texas. He moved to Knoxville.
Right.
Continued as associate professor at the University of Tennessee. Was promoted to full professor, and then he went to LSU in Shreveport, become chair of biochemistry and molecular biology. And then in 1991 went to Hershey, Penn State Hershey, as chair of microbiology.
Which for him, in a way, was going home. So that's nice.
Yeah.
What was his years of life?
He was 77.
70. Oh, my goodness. That's
not very old.
No.
And he was
I'm
quite active. I also met him on several occasions, and I would echo what Rich said. He's a gentleman and a true virology scholar. He, in addition to those chairmanships and so forth that were mentioned, he was quite active in ASV. He was one of the charter members. He was an emeritus member, symposium chair, and like Rich and I, he was program chair. So he was quite in the trenches. He also served on the finance committee. So quite a contributor to the virology field.
Another virologist we've missed on TWiV.
Yep.
Now, I went to the Science March, the March for Science here in New York City this past Saturday. And that was a lot of fun. I was a co-master of ceremonies for our event.
No kidding.
Yeah, here at Washington Square Park. It was very cool to do that. Get up on the podium and introduce people and get the audience all riled up. At one point, I got up and I said, as a professor, I can't help but ask you a question. How many of you currently have a virus infection? There are a few thousand people there. And about 4 raised their hands.
Yeah, right.
I say, wrong. Google me to find out why. Anyway, Justin, a listener of TWiV, wrote, I think I was in front of you for most of the march. Anyway, just noticed now, thanks for supporting the events. He sent a picture. I started talking to a woman who was next to me. So after the event, there was 2 hours of talks in the park. Then we marched from Washington Square Park to Wall Street. And I started talking to this lady who turns out to have been a former English professor at Drew University. She's now living around Columbia, and she's on the Morningside Ayakuk. She's the local neighborhood, person on the committee.
Lay
person, yeah.
Cool.
So we were talking, and she said, I'm going to start listening to TWiV.
Excellent.
Another fan. So that was fun. Now, a few words about the march. This was great because the students and postdocs made posters. They were chanting, right? The whole time we were walking, they were chanting. And, chants like, well, this is the first one that comes to mind. What do we want? Peer review. When do we want it? Now. I don't agree with that, but stuff like that. And they were doing it continuously. And they were great. However, I did not recognize a single person.
Right.
And I can start ripping off names here in New York City, but I won't. Where were you?
Right.
This is our event to show the world our solidarity and our enthusiasm. And can you imagine, we have a few Nobel laureates here in New York City. Man, what if some of them had showed up? What if PIs of all ages had showed up? Not just the students and postdocs. They're great, but come on, folks. I would have gone to D.C. for this if I hadn't been invited to do this. I'm going to do this next year. Man, we got to get out the people. This is not good because it's going to get less and less people every year, and then no one's going to pay attention. The whole point is we have to make statements about the importance of science. So please, PIs, lab heads from young to old, please next year come. It's a great event. You feel so good marching for your field, the field that makes everybody happy. Very cool. All right, a few more things. Anthony sent us a note that April 12th was the anniversary of the Salk vaccine release. It was 1955, I think, right, Kathy?
55. Mm-hmm.
Because I have a pin in a booklet that you gave me when they had a celebration at Michigan for, I think, the 50th anniversary, right? A polio pin. And finally, Eric Carter, a longtime listener of TWiV, visited the Wall of Polio last week.
Yeah.
Dickson met him.
Yep.
He wanted you to sign his Vertical Farm book.
Yep.
And so Eric is working in a lab in Boston. So you can visit the farm, the farm. The wall. There's no farm here.
No.
You can visit the wall this summer. It's a great destination.
That's right.
What are the gold icosahedra?
So those were sent to me by Islam Hussein, who found them at Target. They're just called decorative objects. And so he thought I would like them. And of course, they're lovely icosahedra, which embellish the wall.
And they kind of frame Eric's head. So
They do frame his head. I make tall people. Well, usually it's the short guy who stands in the middle, me. But Dickson said I had to be on the flank. A few more emails. Anthony writes, there's some work using the olfactory bulb. Okay, last episode that we did together was about putting the rabies peptide linked to an siRNA against West Nile into the nose of mice.
Right.
And he gives a link to an article, Sheep's Mucosa Shows the Way to More Effective Medicine for Severe Neurological Diseases. That's the title of the article. Big challenge in science is to get medicine into the brain, and they talk about using the nose to do that via the olfactory bulb. This does not seem to be the case with recreational drug use, where cocaine is absorbed through the mucous membranes lining the nose and sinuses. So, Dickson, you said cocaine would be absorbed there, but apparently it's not.
But that's yeah.
It gets into your blood, which, of course, your mucosa is highly vascularized. Yeah, so there you go. And then
the link that he provided on that gave me a robust education in how to take coke. Okay. So I learned a lot from that. Good luck. That was good.
And responsive mice to aerosolized rabies virus. So he gives a link to an article where they have aerosolized rabies and infected mice. All 4 mice were killed and found to be rabies positive. So it gets in the brain. They develop antibodies.
What kind of facility do you need to carry that experiment out?
Rabies.
Aerosolized rabies.
I wouldn't be surprised if that wasn't VSL4.
I wouldn't either. I would think.
I don't know. It's a good question.
Wow. And how long does rabies last outside a cell?
Don't know. It's got a membrane. Okay, so
right, because when you walk into bat caves, right, and you kick up all that stuff on the floor.
We've talked about people.
And you can
this idea of getting rabies in bat caves, and I talked to someone at University of Georgia.
Yes.
Works on rabies, and he told me that was nonsense.
Nonsense.
You don't get it by aerosol.
Okay, good.
Well, I've talked to people at CDC with the same question. Inger Damon comes to mind, and she didn't say nonsense, but she said there was no evidence to No evidence to support it. And that's what inspires this kind of research, because it is a controversial issue. Well, in the introduction to this paper, one of the things that they point out is that in every case where there's suspicion of infection via aerosol, there are other possible causes, okay? Contact, physical contact with bat secreta of one sort or another, etc. So,
okay.
I think the person you were thinking of at Georgia was Zhen Fu.
Yes, right. Exactly. Yeah, he didn't say nonsense. That was just my word. I tend to embellish.
Do you?
Really? Did you notice?
Just a little.
So he probably said the same thing that Damon said in a very conservative way. There's no evidence that it happens. But Racaniello, containment for rabies virus. Let's see if we can find...
So what is it? Mice die, monkeys exaggerate, and Racaniello embellishes.
Racaniello could embellish. Say, according to the Boston University rabies virus agent information sheet, it's BSL-2.
Huh.
Maybe because there's vaccines.
And they say the transmission is by skin exposure,
Interesting.
Or mucous membrane splash to eyes or nose or mouth. So, you could splash yourself. So just be careful, Dickson, when you work with it.
I will from now on.
Our snippet was sent by Basil, who writes, I hope this makes it to a snippet. I'm sure it will make Dr. Condit intrigued to share his experience with us. A case report of cowpox infection in human, likely delivered by a cat. This is a New England Journal of Medicine. It's really images. I guess they have a series called Images in Clinical Medicine, right?
Yeah.
Cowpox virus infection by Ewa Talarek and Magdalena Marcinska. 11-year-old girl with a skin lesion on her left cheek that had been present for 3 weeks, lived in suburban Poland, hence the two names I had difficulty with, and had direct contact with cats that lived in her home and roamed freely. No bites or scratches. Parents reported initially a pustule had appeared, evolved into a necrotic ulceration with a black eschar, 2 centimeters in diameter, accompanied by expanding local erythema and submandibular lymphadenopathy. Swollen lymph nodes under her jaw.
Correct.
She had been treated with antibiotics, but there was no improvement. Incision did not yield pus. Bacterial culture was negative. Swab were tested for orthopox virus DNA using cowpox virus-specific oligonucleotides, which were positive. So there you go. No treatment. In this patient, healing was prolonged and surgical removal was performed after 3 months. A 1 centimeter scar surrounded by residual area of erythema was present. So this comes from the Medical University of Warsaw. Dr. Condit, that is a nasty-looking lesion.
It is a nasty-looking lesion, and 2 centimeters is no joke.
No.
I think that she didn't actually show up to these people until 3 weeks into this, or something like that, which is a bit of a surprise. Because if I were a parent and not a pox virologist, actually, even if I were, and I saw this going on, it would freak me out. It's a nasty-looking lesion. But, it's classic. This is a cowpox infection, and it's contained. It's just this one lesion. That's it.
Mm-hmm.
And then it goes away.
Is that typical to have just one lesion?
Yep. Every now, that's just like a smallpox vaccination. And every now and then, in particular, if you scratch it or mess around with it, you can get satellite lesions, usually because you physically transferred, because it'll have live virus in it, physically transferred the stuff from one site to another. But typically, just one lesion. That's a whopper.
Rich, is there a scar afterwards?
Yeah, probably, just like your smallpox vaccination.
Yeah, exactly.
Scar.
So this is on her cheek.
All right, so how did she get this?
This is on her cheek.
How did she get this?
So, the supposition here, I think it says here that she has a cat.
Yeah, cats.
And the cats are not, I don't think the cats are what I would call an active host. They're almost like a fomite. Okay. The reservoir is going to be rodents, and cats interact with rodents. And my guess is that they carry the virus around on them, a cat messing with an infected rodent, and you snuggle with a cat and bingo. Now, what they do note here that she had no scratches or anything, but all it takes is the tiniest break in the skin, and a high enough dose of virus, and that'll launch the infection. And then
so maybe the fur had virus. She got it on her face. Maybe she just slightly scratched her face, and that was enough to do it.
Something like that, or maybe she had some other pre-existing lesion on her face, like a blemish of some sort.
Now,
And,
Rich, there's no in the U.S. Would you be able to get an antiviral for this?
That's a really outstanding question. In an emergency, there are two good anti-pox viral drugs. One is ST246, and the other is CMX001, which is a cidofovir derivative. That's a nucleoside analog, and the ST246 is entirely off in its own thing. It interacts with a process in a poxvirus infection that's part of assembly and spread. Actually, I don't know about CMX001. I don't think that is actually formally FDA approved. Cidofovir is, but I don't know. This is a matter of me not knowing whether or not this derivative CMX001 is approved.
But 246 is approved. Yeah,
it is not approved.
Oh, okay.
Okay. That process is still ongoing. They're still working towards approval, and this gets into how do you evaluate a drug where you can't do the experiments in humans? And this is where we come to the animal rule for identifying appropriate animal models and stuff. And so the company that is developing ST246 and the government have been dancing this dance to figure this out, and we'll see what happens to that. However, they are, in fact, if I'm not mistaken, stockpiling ST246. And in emergency situations, and there have been several of them, you can get this. I forget what the official name is for the process that allows you to get a drug that is not FDA approved in an emergency situation. And I don't know how much of an emergency it has to be, whether it has to be life-threatening or not. But definitely in a situation where a patient's life is at risk, you can get CMX001 and ST246, and typically they also still give gamma globulin, sort of passive immunization with something that they presume has some antibodies in it. But it would be great to see a real human trial somehow with ST246, because I'll bet you the stuff works great. But
you can't,
you can't do it. There's not enough.
Yeah. But what if
not enough cases? You can't do a controlled trial.
There's some countries in Africa where you get monkeypox, right? And maybe they could test it there.
That would be interesting.
Rich, what happens if this? I'm sorry. Go ahead.
Go ahead.
What happens if this virus gets into your eye?
You can get an infection in your eye, and
of course. But I mean, is it sight-threatening?
I think it can be, but usually I believe it clears up. I'm not omniscient on this, but I do know of several cases. As a matter of fact, I have personal acquaintance with people who have had infections in their eyes. And in all cases that I'm aware of, it's cleared up.
Right.
And there's been no long-term sequelae.
No, that's really
I should say that one of the typical sequelae of a smallpox infection for people who survive the infection was blindness. Okay.
Interesting.
So smallpox at least can blind you.
Yeah, sure.
4 years ago, Rich and I interviewed Dennis Ruby, one of the people who's been involved in the development of antivirals, right?
Yes.
Smallpox.
He's, as a matter of fact, the company that he works for, SIGA, is the company that has moved ST246 forward.
Right.
It was originally discovered and researched in a small company called ViroPharma. subsequently went out of business. And Dennis took I don't know whether you have to purchase licensing and all this kind of stuff, but basically he took over the project in SIGA. And in fact, hired the scientist who did the initial discovery work, Robert Jordan, who worked with Dennis in SIGA for a long time and did the most of the bench work to bring this drug forward. And they've done an outstanding job with it. They've not just done 2 animal models. They've tested many animal models, and it's, from my understanding, efficacious in all of them. And they have been able to do, you can do toxicity and pharmacokinetics trials in humans. Okay, so you can give humans what you think is an appropriate dose of this and ask whether or not they get sick, whether there's any side effects of the drug and how it's metabolized. And that kind of stuff has been done. And in none of the animal models nor in humans are there any toxic effects of the drug. So, for all intents and purposes, it looks good.
TWiV 270. Check it out. He talked.
Remember that? Where were we? We were at some biodefense meeting.
Yeah, ASM Biodefense in Washington.
Right.
I don't know if
this has been going on for a long time. When was TWiV 270?
4 years ago.
Yeah, and this has been going on for much longer than that.
It was the half-life of TWiV.
Yeah. All right, we have a paper also suggested by a listener. Dear TWiV, I wanted to bring your attention a recent publication authored by Greg Stewart and myself that describes an interesting virus that encodes fermentation genes. Entitled "A Giant Virus Infecting Green Algae Encodes Key Fermentation Genes." It's open access, provides a link. Thank you for your great work every week. Christopher Schwartz and Greg Stewart from the Department of Oceanography at the University of Hawaii in Honolulu.
I want his job.
Yes.
If I had it to do over again, I'd get his job.
Well, they just recently evacuated a lot of people out there, so take it from that.
It is in Honolulu, but it's the University of Hawaii at Manoa.
Yep. Where's Manoa?
Somewhere near Honolulu, I guess.
Because the address says 1950 East-West Road. That's a great Honolulu, Hawaii.
That's right.
So I like this because first, one of the authors sends it in, and if you have the good taste to send a paper to TWiV that you have yourself authored, ye shall be rewarded. If it's a good paper, that is. And of course, you won't send it to us unless it is. This is the isolation of another mimivirus, but it's got interesting properties, as you could tell from the title. And I like it for a number of reasons, but one of which it was isolated from the coastal waters of Oahu. And the host from seawater 100 kilometers north at the Hawaii Ocean Time Series Site Station Aloha. It just makes me think of Hawaii.
True.
And it's a mimivirus, and we'll talk about it. And I want to tell you how they got it first. And Rich Condit has actually corresponded with the author, so he can tell us all about that. The virus was isolated from surface seawater, as I said, collected from the Oahu side pier to Coconut Island. Now I'm getting hungry. Located in Kaneho Bay, Oahu, on September 2nd, 2010. This is almost 8 years ago. 175 liters of surface seawater. Then collected, they brought it back to the university where they filtered it through 0.8 micron filters, 142 millimeters in diameter. So big filters, right?
That's 142 millimeters. Those are the kind of filters on a stand that we used to use to filter media. Is that right? Yeah. And you'd filter, 20 liters of media and then pull the filter off and it had cracked and you had to do it all over again, right?
So they filtered it. They then concentrated it.
0.8 microns is important because ordinarily, if you want to filter out bacteria and maybe let most of the viruses through, you might use 0.2 microns or something like that. But they were interested in not excluding from this giant viruses. So they used a larger pore size that'll get out most of the non-viral material, but let the big viruses through.
Yeah. So the question to which I don't have an answer: what life forms will pass through that 0.8 micron filter? Dickson, do you know?
All the life forms that will pass through are smaller than 0.8, and all the ones that
Thank you so
much. You wanted a direct answer to the question.
Names of the organisms.
Well, the names. I'm not a marine biologist. I can't do that.
You're a biologist, aren't you?
Yeah, I am, but I can't.
See, I'm not. I'm a virologist.
Okay, if you start from like protozoans and go down, you can say all the protozoans will stick. They will be filtered.
What about these algae?
Algae will stick.
They're bigger than 0.8 micron.
Oh, yeah.
Okay. So not a lot of.
I mean, for size reference, a red blood cell is 7 microns in diameter.
Okay.
So you can put it in context then.
How big is a lymphocyte, Dickson?
It's bigger than 7.
Okay, that was an irrelevant question.
It's about 38 microns.
They then they used this concentrate to challenge tetraselmis cultures.
Selmis, you were right the first time.
Selmis, you were right the first time, yeah.
I'm looking at
Selmises.
I'm looking at a typo in the manuscript where it says slemis.
Oh.
In the methods.
Well, I'm looking at the abstract.
Slemis. It's wrong. Selmis. Yeah.
That's right.
And Rich will tell us in a moment why they picked tetraselmis.
Right.
But then it grew, and in fact, it grows lytically in tetraselmis. And then they isolated the virus by 3 rounds of dilution to extinction in 96 well plates, which means you keep diluting until you don't get any more CPE, and then you back up, right?
Yeah.
Right.
That's it. So they say the host of this is tetraselmis, which is a
Chlorodendrophyceae.
A green algae.
Actually, I should have asked him how they know that the cells are infected. You said CPE, but this is a green algae. Does it have a cell wall?
It says it results in cell lysis.
It does have a cell wall.
Okay.
All plants have cell walls.
So that's the cytopathic effect in that case is lysis.
Yes.
Yeah, that's how they know. I assume they don't have a plaque assay for this.
No.
I just wanted to add that tetraselmis is a genus, so there are a number of species in this genus.
Yeah, so they used, I think, tetraselmis cordiformis and others. Yeah, they tried multiple isolates. They also tried some other chlorophytes. So, Dickson, what is a chlorophyte?
It's an algae that contains chlorophyll. Chloroplasts. They have chloroplasts.
So these are algae living in, they say, both salt and fresh water, but they live near the surface, the photic zone, right?
Exactly. They would have to.
I'm picking up these cool words.
They would have to.
Are they benthic, Dickson?
No, they're not. They will eventually end up there, though. As we all end up there.
Are they riparian?
No.
Yeah.
Are they estuarian? And the answer is yes.
Exactly what I was going to say.
I was because I pasted these maps in, and the listeners won't know about this yet. But Manoa looks like kind of a neighborhood in Honolulu, just north of H1, and where they collected the seawater off of coastal Oahu is Kaneohe. I don't know how you say it, Bay, but that's on the other side of the island.
Over Diamond Head?
Yes. If you look at something, I put that map in as well.
Good.
And then another point of geographic reference is about 100 miles north of Oahu, right? Or kilometers, 100 kilometers north is where the seawater was collected at the Aloha Station.
Kaneohe Bay. If you've been, it's great snorkeling.
And if you go way up to the northern tip, they got a great shrimp truck and a good shave ice truck as well.
The shave ice truck at the north is great. Yeah. It's the best I've ever had.
Yeah.
Absolutely.
Yeah.
Yeah, it's pretty cool. Nice place to work.
I'll say.
Except, like I said, right now they're suffering from floods.
Mm-hmm.
They've got a huge amount of water.
So they have an electron micrograph of an infected tetraselmis cell.
Yeah,
so they're roughly spherical.
Right? Almost like the outer capsid looks like benzene.
Yeah, it's icosahedral, obviously, but the cells are spherical, and these are photosynthesizing algae, right? Which
yes,
is
they are.
All algae are,
Green,
right?
Green.
They're green.
Yeah.
And they take light, Dickson.
They do.
And they make carbohydrates.
They do that.
So
via the Hill reaction.
They need to be near the surface, but if they're not, what happens if they drop down? Do they get sick?
Well, you know what?
Can they move? They have flagella, right?
Yes, they have flagella.
When they do, but when the waves move, there is a countercurrent current per opposite waves, and it actually keeps the algae in suspension at the photic zone.
Wow.
Which is a remarkable aspect of the motion of water.
This photo they have here, and this is open access, so it's virology, by the way. You can see it. It's lovely. It's full of particles.
Yes, I mean
a lot of particles. It's very clear that it's infected. Although it would be nice to have an uninfected cell so that we would believe them, right? That this is not what an infected. But they don't. So we'll take their word for it.
Right.
So this thing does have a cell wall, right?
Of course.
Is that what I'm seeing? And so
yeah, it does.
The virus has to burrow through there
somehow
to infect these guys somehow.
That's very interesting.
Although it has a flagellum, so it could enter through the flagellar pocket.
Really?
Ooh.
You mean where the flagella goes through the cell wall? Yeah.
Correct. That is correct.
It's true. That's
trypanosomes have that deal, by the way.
Right. So they sequenced the genome of this virus.
And one of the things that makes this special is that with one exception, all previous mimiviruses have been had amoeba as hosts.
That's right.
So we're talking about a new host range here. Right.
Now, Rich, you and I met Grieg at the big virus meeting in Germany. You didn't go.
I wasn't there.
Oh, you weren't
Remember?
there. Okay, so he was there.
I couldn't go to that meeting, unfortunately.
Yeah, right. You were sailing.
Boy, but I'll tell you, I'm about to get on a plane and go to Hawaii. Just to have a conversation and lunch with Grieg or something.
That's a great idea, Rich.
That sounds so.
So tell us what you had a little conversation with him about. This. What were you interested in learning?
We're kind of getting there, but the way this paper is presented, it's presented with basically the unspoken, really undirectly spoken conclusion that this tetraselmis is the natural host of this virus, which they call TEV1, tetraselmis virus 1.
Right.
TEV1, that's right.
And my question to him is, I kind of, as we'll get into this, we'll see that they tested for the growth of the virus on a bunch of different hosts and that they had available, and this is the only one it grew on. It's got genes that are homologs to genes from this particular host. So there's a lot of circumstantial evidence that this would be the host. But my question to him is, you don't really have any formal proof that this was that this is the natural host. And he said, absolutely right. We don't have any formal proof that this is the actual host. Okay. But for all the reasons that I've already said, this is the only thing that they've got that it grows on. It looks like other viruses that grow on blue-green, on green algae. It's got genes that match the genome of the algae. It's a good bet. And then I backed off and I said, so how did you come about with this host pathogen pair? Okay. And it turns out that this is a part of, I think, a much larger study that I think we'll hear about in the future, where they took this 175 liters of seawater, concentrated, so they have a virus stock, and tested that to see if they could see infection on a number of different organisms. And this tetraselmis is one of the ones that came out of that. And so now they had a good host pathogen pair, and they went ahead and did the analysis, and the subsequent analysis that we'll see that I've already previewed supports the notion that it's the natural host. But they've got a lot of other stuff coming out. I think they have other viruses that they can culture, and then, of course, they can do sequencing on the viruses in their seawater. So this is basically a virus discovery project, and we'll see more of it down the road.
Rich, what would you accept as evidence that this is the natural host for this virus?
I'm asking myself the same question, actually.
I mean, isolating an infected
that's what I'd like. I'd like to.
That's got the virus inside, right?
Sure.
But I mean, that would be tough. I mean, there's no biology in this paper. That actually would have been nice to see some sort of a time course of virus infection or something like that.
Sure.
So it's hard to know what the probability would be of being able to isolate one of these cells and see it infected. But I mean, ultimately, you'd like to see a virus-infected cell isolated from the wild.
I volunteer to go look.
No, me.
Good idea. I'll go with you.
No, me.
So we can just get water every day.
That's right.
Yeah.
We can go in the morning and we can, get these tetraselmuses. We can put them in the incubator. Then we can go have lunch.
Have some
then
shrimp.
we could go snorkeling and then go back and check the incubation.
Right.
Okay. I think it depends.
While things are incubating, you need to be lying on the beach.
The question that you alluded to, Rich, is if you infect a culture, how many cells get infected, right?
Right.
Like what's the efficiency? Is it everybody if you use a high MOI, or is it very inefficient? Because if it's everybody, then maybe you could find infected viruses in the ocean. So
and in the
cells.
in the ocean, where it's dilute and everything else, maybe there'll be 1 in 1,000, 1 in 10,000 of these guys that are infected, and if the infection lasts for 24 hours, it's going to be pretty transient. So the odds of finding an infected cell in nature are pretty slim.
It's okay. I'm willing to spend some time.
I was going to say, we're up to the task.
No problem.
We are patient people.
We can bring a microphone and a recorder. We can do our podcast there.
We could do that.
And the beach sounds like a good idea. Maybe we can investigate other viral niches.
I understand the bone fishing in Hawaii, right in Honolulu itself, as you look at Diamond Head, is spectacular.
I actually would like to do this on the other islands, not Oahu.
Well, you'd have to do a survey, of course. I mean, you can't just
some of the other
island.
phytoplankton they looked at, where they collected from this Station Aloha, which is where the tetraselmus come from, and also Kaneohe Bay, which is where the great scuba is. Chlorophytes, Pyramimonas, Micromonas, and the parasinophyte clade 7, as well as other photosynthetic flagellates such as arachneophytes, cryptophytes, dictyocophytes, and haptotypes, and pelagotypes.
Pelagotypes are open ocean types.
None of them make any recognition in me, but says these are not words, but I do recognize.
They're a little like angiosperms.
But they're all negative except tetraselmus. So
right.
I don't know what else is out there. Yeah.
I just want to interject. If we go, we're going to probably not want to eat salads where the lettuce hasn't been washed really well.
Here.
Absolutely. You've been listening to TWiV. Yep. I said, I'm not eating salads there. No way.
Was that listeria outbreak?
No, that's the rat lungworm that one patient got from having a salad.
Oh, sure.
In Hawaii.
Sure, absolutely.
It wasn't proven that it was the salad.
Yeah, it was.
That was the
pretty good.
Likely culprit.
Yeah. We could steam our lettuce. Oh, that would be lovely.
Yeah.
You.
I'll take the risk. I'm going to go for it. I like salad.
Rich.
This person who had this lungworm had a big problem.
Yes, she did.
You have long-term neurological issues. You can't get it out. It goes in the brain, and you can't get it out.
You don't want this one.
All right, so they sequenced the genome, double-stranded circular DNA, 668,031 base pairs, which they say is the largest genome for a virus infecting a photosynthetic organism. You could also get more restricted if you wanted to, but you don't have to. Right. And what's interesting, 663 predicted genes, 10 tRNAs, okay? But what I think is cool is that 67% of those open reading frames, which is 435, have no similarity to known proteins.
It's incredible.
This is just one virus that they plucked from the sea.
Incredible.
And just keep doing this.
Shows you what we don't know.
Yeah, exactly.
Is this a known unknown?
A very known unknown.
43 of the 110. So there are some that look clearly eukaryotic, 110, and 43 are from the group Viridiplanti, which includes chlorophytes. And so that's what Rich said earlier. It looks like some of them, 19, are from Tetraselmis. They seem to have been inherited or are highly related to Tetraselmis genes, which is part of the evidence that he thinks that this is a host, right?
Right.
Now, it does have the smallest, it's not the biggest mimivirus genome. It's not the smallest. So 683,000, years ago would have been cool, 668,000, but nowadays, what's the biggest mimivirus? We can look that up, giantvirus.org. Giant
Is that right? Giantvirus.org?
It's on the tip of my tongue. Yep. And here we are, top 100 largest viral genomes. So the biggest mimivirus is mimivirus, which is 1.1 million bases. Yeah.
So that's almost twice the size of this thing.
Yeah. In the acanthamoeba host?
Yes, these are pretty much all amoeba except for the Cafeteria rowenbergensis virus and the Emiliana huxleyi virus.
That's a very interesting organism.
It's a Cheshire cat. Yeah, it's very interesting.
Amelia Huxley is
we did do a TWiV ages ago. Or maybe that was TWIM. Okay, so that's the genome, and there's lots of unknown genes. 192, only 192 could be annotated, so they use those to try and do phylogenetic analysis of this. What I find, and they look at a number of different genes that they can identify. There are 4 genes in this virus that have never been seen in any other viral genome. And I think those are the ones we can chat about, although we can, of course, chat about whatever we want because no one's telling us to stop.
It's our show.
It's our show. But the first one, which I found very interesting was, 2 of them, pyruvate formate lyase and pyruvate formate lyase activating enzymes. And so I looked these up as they are not part of my lexicon, which is very small. Pyruvate formate lyase helps regulate anaerobic glucose metabolism. It catalyzes the conversion of pyruvate and coenzyme A to formate an acetyl-CoA. And of course, acetyl-CoA goes into the Krebs cycle, and that makes energy.
Into fatty acid synthesis.
Or fatty acids, and that makes, either way, you can get energy out of that.
Exactly.
And the other enzyme, pyruvate formate lyase activating enzyme, activates the pyruvate formate lyase.
Right.
So that enzyme has to be activated before it can do its conversion.
Yep.
So they say that this could, it could allow the green algae to ferment when
quicker.
All right. So they say during periods of low oxygen concentration.
Yeah.
So you got sunlight, you're doing photosynthesis, but you're producing oxygen.
You are.
So what do you need oxygen for? Other parts of the metabolism?
Algae have mitochondria as well.
Yeah, so for that. But could you live without oxygen for any period of time? Just photosynthesize? I guess not, right?
No. They have to go at night.
Brief periods of time. So they say brief periods of times low oxygen, and they say especially at night.
Yeah.
Or in areas where there's a lot of nutrients and you get a lot of growth of other things.
Yeah, you
and
get
they take
oxygen
away
depleting.
Anoxic.
That's
right?
correct. Agricultural runoff, for instance.
So they say maybe these genes are important during that time. But of course, the host has these genes as well.
And by the way, agricultural runoff is what's going on right now in Hawaii.
Yeah.
And who's the agriculture? Dole pineapple.
They should Can you grow pineapples indoors?
Yeah, sure. You could grow anything indoors, remember?
No, you can't grow an apple tree indoors.
Yes, you can.
It's too many. It's too much.
Well, I didn't say don't do it. I would say don't do that, but you can do it if you want.
So, Rich and Kathy, do you know if the Tetraselmis has these pyruvate enzymes in them? I didn't look it up.
I did not look it up, but I believe they do.
If you look at Table 1, pyruvate formate lyase activating enzyme, blast top hit seems to be Tetraselmis.
It is.
Yeah, good.
Now,
and the lyase is in a clammy Chlamydomonas.
How close is the viral pyruvate formate lyase to the tetraselmis pyruvate formate lyase?
They probably have that here, but I don't remember. But
I mean, was one derived from the other?
Wait, you're just asking about the viral one and the
cellular
host one?
Yeah.
They don't, in this table, they don't show the top hit as being from tetraselmis. They show it as being from Chlamydomonas.
Oh.
But maybe it's in tetraselmis also.
I see what you mean.
Since the activating enzyme is in tetraselmis.
No, in table 1, you read across. It's TWiV 456, right? You've also got tetraselmis in the next column. Right?
Yeah, for the activating enzyme.
For the activating enzyme, not for the lyase that you were asking.
Why do you
about.
want to know?
Lyase activating it. I just want to know if the virus stole the enzyme from their host.
Did.
Yeah.
Well, then you could find out if this is the host or not.
That was one of their supporting pieces of evidence that this is the host. Yeah.
If you go back to Figure 3, Dickson, where they talk about homologies, there's 19 of the genes. One of these must be the formate lyase. 19 of the genes in the virus have sufficiently close homology to tetraselmis so that they look like tetraselmis genes. Mm-hmm. And yes, that is used as evidence that this is the host.
Now, how convincing is that, Rich? Just give me your opinion on that.
I'm okay with it.
Okay.
All of the evidence put together.
Yeah.
I in particular like the host range studies.
Mm-hmm.
Okay. But the host range studies and the virus apparently copying genes from this bug is convincing, sufficiently convincing to me. So this is great, as Greg says in his letter, I'm perfectly happy to be surprised.
Good. That's
Science
great.
is full of surprises.
That's good.
I want to back off just a little bit here and say that what's unusual about this, that this is a selling point of the paper, is that this is, you said that these are genes that have not been found in any other virus, and in more of a general sense, this is the first example that, so they say, and first example that I'm aware of viruses having genes that tinker with energy metabolism, intermediary metabolism in the cells.
Or at least potentially, because we don't even know if these genes are expressed. Right?
You remember
that's right.
About TWiV 2 or 3, when Vincent and I were just talking, and he asked me to describe what I thought a virus was, and I said it's an organism, it's an entity that doesn't have any metabolism.
Right.
And this might contradict that.
It's got 2 enzymes. Does that constitute metabolism?
Well,
it can tinker with it.
So here's the deal. Does this offer an advantage to the viral replication cycle by increasing the energy metabolism of the host?
That's exactly what they post. And that's what you could do. You could delete the gene and see what the effect is.
You could do that or mutate it so it doesn't work.
Well, yeah.
If you could do genetics on this virus and the host and that kind of stuff, I mean, yes, that theoretically you can do that, but that's a long way down the road. And more particularly, it's from the characteristics of these enzymes, it's like tinkering with the metabolism under circumstances of oxygen deprivation.
Which you could do in the lab.
Which is why they called it fermentation. That's exactly.
You could do that in the lab.
You could. And algae are really easy to grow in the lab.
Actually, I suppose you could try and do these sorts of experiments with, RNA knockdowns, antisense RNA knockdowns.
Can I start working on this?
Hey,
yeah, but you'll have to move to Hawaii.
Oh, I can get to it. You could send me the virus and the algae. I could grow it here in tanks.
I think you should go to Hawaii.
Good.
I would like to start something new, and this is exciting. Develop a genetic system for this. Probably there's no money. NIH won't fund it. It's probably an NSF thing, and they don't have enough money.
Well, it's a good thing.
I'll just keep podcasting. The other enzyme is mannitol phosphate dehydrogenase.
Right.
It degrades mannitol. What it does is it makes from mannitol fructose 6-phosphate. So they say that there's a lot of galactose and galacturonic acid in the cell wall of tetraselmis. So maybe this I'm sorry, I'm mixing up two different things. The what enzyme did I just say? Mannitol phosphate dehydrogenase.
Yes.
Yeah, that breaks down mannitol, and they say that may alter the osmotic balance of the interior. Maybe it helps the virus to lyse the cell. Forced production of mannitol could induce hypotonic shock, which would facilitate cell lysis. So that's that. And then the other enzyme is alpha-galactosidase, which they say may help break down the cell wall of the algae. Right.
For lysis.
For lysis. So those are two things you could test also. And there's one more, right? Is there? Did we do? No, that's 4. Those are the 4.
4.
Mm-hmm.
Those are the 4.
So those are. So yeah, it's not easy. There probably isn't a genetic system for this organism, but I'm thinking CRISPR. Put CRISPR in it. But you got to get it through the cell wall. So you first have to make without the cell wall. What do you call the cells?
Spheroplasts.
Spheroplasts.
Well, at least that's what they're.
No, you're right.
I think that's
right.
what they're called.
Right.
So you take a spheroplast. You can put nucleic
bacteria
acid
without
into
a cell wall.
So that's what you do with plants. I think you get spheroplast. You put nucleic acid in, then you grow it up into a plant.
Yeah.
So
plants, they call them protoplasts.
Protoplasts.
That's right. There are some ways of shotgunning it through too, but that's clumsy.
The reason I say I would like to work on it because it's BSL -7. You don't have to worry about anything.
That's true.
And it would be fun to grow green cells, develop a plaque acid. I seem to remember Jim Van Etten having these green monolayers, right?
He didn't have to stain his monolayers because
with plaques and
the host organism is green itself and it would form a monolayer. Yeah. You stick the chlorella virus on those monolayers and form plaques. You didn't even have to stain them. They showed right up.
Yep.
Because in this paper, there are like at least 10 different questions that immediately pop to the surface. And I think it would be fun to do something different. This has some appeal. I won't do it, but in science, you do have the flexibility to change.
You do.
Mm-hmm.
As long as eventually you can get funding for it. Of course, here at a medical center, they may say, we're not really interested in viruses that infect algae, but maybe something good comes of it. Who knows? Medically relevant. I like the word they use in this paper, mimivirids.
I wrote beside that, is that even a word?
So, Kathy, in another episode, we talked about this, and then Jens Kuhn wrote in that is how you call the members of a family.
I forgot.
Virids.
Okay.
Virids. So, yeah. But this is the first time I've seen mimivirids.
Mm-hmm.
Can we use that for a title? Green mimivirids. They're not really green, I guess.
No.
So anyway, Christopher, thank you very much. Hope we did justice to your paper.
This, by the way, is part of Christopher's PhD thesis.
Nice.
One other thing stood out for me. They say the largest hypothetical protein from the genome is 8,212 amino acids long. And if you do the math, that is a 900,000 Dalton protein.
That's big.
That's a monster.
You think?
I wonder what that thing does.
I wonder if it's processed.
I wonder if it's tetramers or quadruplemers or whatever.
Huge.
Yeah, and how many of them fit inside this thing?
How many angels can you put on the head of a pin anyway?
Maybe they're not in the virion.
Right.
But yeah.
Okay, thank you, Christopher and Grieg, and enjoy Hawaii.
And be looking out for us,
I was invited to this. They had a giant virus meeting in November again. I couldn't make it. I was invited. Now I'm really missing it. Hopefully I can go back.
The giant viruses are really cool. They're fun to follow.
They are. They're cool. All right, some email. Darren writes, regarding viral piRNAs in other animals, this preprint of ours might be of some interest. This was something we talked about a while ago. And this is a bioRxiv preprint. Metagenomic sequencing suggests arthropod antiviral RNA interference is highly derived and identifies novel viral small RNAs in a mollusk and a brown alga. Cool. Now, Dickson, brown algae don't do photosynthesis, right?
I wouldn't say that.
Because they're brown and have chlorophyll?
All the algae do it.
Yeah?
All of them. So
these don't have chlorophyll.
No, they had another kind of...
They have a different chromophyle.
Yeah, xanthophylls and things like this. Chromophylls, they call them.
God, your knowledge is expansive.
Thank you. I am here to serve.
This is pretty cool.
I will give you a Siri answer for this.
So,
we are here to serve.
Does anyone remember where we what we talked about pico RNAs? What paper was that? That's like over 3-4 weeks ago. Let me look at the listing here. And
fairly recent.
It was pretty recent. I'm thinking it has something to do with mosquitoes. Yeah, I think so.
Yeah, I don't think I was even on the episode. I think I remember hearing about it.
Let's see, Milwaukee, Sean Whalen, fishing with defective flies. Nope, that's the defective, but that was also insects. Mimivirus and bats. No, that's the single. Don't evome without antibody expressed. That's endogenous viral elements. I don't think that was it. Biles to go before I delete. I don't think that was it. All right, it's too far back. Anyway, this looks like a cool paper. We will have a look at it, and we'll have maybe when it gets reviewed and published. Now I have lost my show notes. Okay, here they are back. Rich, can you take Clarissa's email, please?
Sure. Clarissa writes, "Hi, Vincent. I sent these links to Rich, and he suggested that I should send them to you/TWiV. I don't know if you've seen these papers that appeared in M-Sphere about horsepox synthesis." This refers to the paper we did a while ago from the Evans Laboratory where they synthesized horsepox from synthetic DNA. Best regards, Clarissa. P.S. Congratulations to the TWiV team. I really enjoyed TWiV 478. I forget what.
That's the one. That's the one.
That's the horsepox one.
Yeah.
Okay. Excuse me. Little cough time out there.
Nope.
And she includes a link to Mike Imperialis. He's the editor of M-Sphere. He's editor-in-chief.
Editor-in-chief, yeah.
Commentary on why M-Sphere is doing this, and he writes a nice little overview saying that we need to air all sides of these issues. And then there are two articles, one from Greg Koblenz, titled, I'll get it here, Critical Analysis of the Scientific and Commercial Rationales for the De Novo Synthesis of Horsepox Virus. And the other from, let me get the actual authors' names, from Diane DeEulis and Gigi Quick-Cornval, called A Holistic Assessment of the Risks and Benefits of Synthesis of Horsepox Virus. And these two articles kind of, sort of take both a pro and a con view of this. The con view being Koblenz's article that's a critical analysis of the scientific commercial rationales for the de novo synthesis of horsepox. The holistic assessment, the other article, in fact, doesn't offer a lot of real opinion about whether this is a good thing or a bad thing, but does a sort of an overall analysis and doesn't take great objection to it. And in fact, says that a lot of the stuff, is really okay. I was interested as much as anything else I always look up the biographies of these people, the CVs. I want to find out who's writing these. And Greg Koblenz, I linked his bio into this. He's on the faculty. He's in the Schar School of Policy and Government at George Mason University, and his background is in public policy. I think he's got as a public policy wonk, a PhD in political science from MIT and an MPP, that's probably a Master of Public Policy or something, from the JFK School of Government at Harvard. So that's those are, impressive. As a policy person, impressive credentials. Note, however,
you should run for office.
he's not spent any time at the bench.
No,
he's not. He has no direct experience in biological science. By contrast, the authors of the other two articles, have training. One is a neuroscientist and the other is an immunologist, and both have morphed into policy positions in government. And in particular, I was particularly impressed with the credentials of Gigi Quick-Gronval, who's worked in the White House and the Department of Defense. She's written she's an expert in particular in synthetic biology. She's the author of a book called Synthetic Biology, Safety, Security, and Promise, published in 2016, etc. My analysis of these, this is interesting, my analysis of these articles, the first one by Koblenz, he spends all of his time basically saying that the rationale for doing the experiments is weak. And the basic rationale for doing the experiments, as articulated in the paper, is the possibility that we would need a new smallpox vaccine, and that since horsepox may have been the original vaccine anyway, and this is the only horsepox around, and it wasn't available, that synthesizing this horsepox is an inroad into making a new vaccine. And in fact, his analysis of that is very well informed and very well articulated, but it doesn't really, frankly, I would not be at all surprised if we never researched the government never invested in making a new smallpox vaccine. We are pretty well fit out for smallpox vaccines. But to me, that's not the point, okay? The interesting thing about this is he doesn't talk at all about he talks about the rationale. He doesn't really talk about the risks, okay? And in my mind, there really are none to this. And under those circumstances, I mean, yeah, you got to give a rationale for do this. And this is what the company wanted to do. And the company basically gave the project, needed a scientist that could do this and gave it to David to do. And David was interested in doing it. Part of me says that really the real motivation for this ultimately is just basic science.
Yep.
Okay. This is a technology that in fact could be very powerful for good, which is one of the things the other authors point out. How many hours have we spent talking about the benefit of just doing basic science? Okay. Yep. Basic science all by itself, fundamental investigations have enormous benefit for society. And I think that that's where this comes from. And I don't think, that there's really any substantial risk, as we've already discussed. It was, in air quotes, obvious that this could be done. This is a proof of principle. So I don't see a great risk to this technology, and I see a great benefit just in the fundamental science. And so to me, all of this talk, however rational and well articulated it might be about whether or not the justification of making a new vaccine is a good justification, misses the point.
Do you think, Rich, that's because he's not a scientist and not thinking that way?
Well, this is kind of where I was going with this. I've been thinking about this over the last couple of days as I've been studying this. And I think that maybe it's, the appreciation for the benefit of fundamental science, really fundamental science, is subtle. And I think maybe it's something that you have to really experience for yourself to understand the magic of just doing an experiment to see what happens, okay? Just out of raw curiosity to see where it goes, and also to be immersed in that culture for a lifetime and understand from direct experience the incredible good and magic that comes out of those fundamental studies. And he's not had that experience.
Yeah.
His experience Now, likewise, I don't know what it's like to be in public policy, but my guess is that there's a lot of, trying to paint things black and white and us and them stuff. So you're looking at conflict all the time. It's a totally different discipline. So yeah, it's a different mindset. And I think the training as a scientist that the other authors have and that we have cultivates an appreciation for the good in these experiments. At the same time, I want to be really careful here because I don't want to. We shouldn't pull play the elitist card here.
Of course,
we got to be really careful not to be saying, "Oh, well, you're not a scientist, so you don't get it." All right, which is, I guess, kind of what I'm saying, but that's dangerous territory,
Richard,
because that sets the scientists apart as kind of elitists or something like that, whereas our role should be to help people appreciate this.
Okay, Rich, I'm going to put you in the room with this guy. How do you talk to him?
In fact, he's very well informed. I don't know. You just have the conversation.
Does he listen to what you just said and said, "Oh, I get it now"? Or,
who knows?
You're not telling me anything I don't know because I spent 4 years at MIT getting a PhD in this subject. So, to be honest with you,
I think we both could have a lot to learn from each other.
Right.
Okay. So I think it would be a very interesting conversation.
When does that happen?
When does it happen? I don't know.
No, not you and him. No,
but that's what we
being
do.
More generalistic.
That's why we do TWiV in part.
But they may not even listen to this.
Yeah, I know they don't. But to try and convey what Rich just said, the awe and amazement of science that we have, and that maybe a lot of people don't have. And I think a number of listeners have picked it up from what we do.
I agree. But I think there's a public fear factor.
Yeah, for sure.
With regards to scientists. And scientists don't do a good job of depressurizing that in public. I mean, we appreciate what we can do and everything, but when you start to talk about this,
I think we're doing public things. Aren't we?
Yeah, of course. But again, I'm going to say that most public people wouldn't be listening to us, unfortunately.
So what do you do then?
Well, that's the point. And so you have this technical meeting like ASV or something like this. Have you ever had policy people at this meeting?
Yeah, we do. So let's say he went to biodefense, right? Just theory. He would go, give a talk, and then leave.
Right.
He wouldn't listen to us.
No, you see, that's not the idea. You have a panel with mixed people.
Well, we could get him on the show and talk to him.
We could.
Oh, that'd be interesting.
We could do that.
Sure. Greg Koblenz. It will be nice.
Yeah.
We won't be arrogant.
He's got awesome credentials.
I mean, he's researched this quite well. I think the one sort of mistake in this that he makes, he's got a rap about how horsepox, the idea is that horsepox would be a more attenuated virus than vaccinia and therefore might be a better vaccine. And he neglects and he just talks about the genome and the genes and says that there's not a good argument for that, but he neglects the biology in the paper where they show that it makes wimpy plaques and it doesn't do much to mice relative to vaccinia.
Right. So
it does, in fact, look attenuated. Yeah. But I think, the rest of the thing is, as I said, well-researched and well-articulated, and I'll bet he's right that this never evolves into a vaccine platform. All right, just as a bet. I mean,
but
that doesn't mean that doesn't matter.
Yep.
Okay, it doesn't mean that in the absence of any significant risk, which is where I stand on this, it doesn't mean that the experiments aren't worth doing.
Yeah, I agree, and I think that if you just said to him, you have to be curious and things will happen, he'll say, that's ridiculous. And then you give him 20 examples of how accidental findings have led to big things. That's what we
The first thing you do is you ask him for his cell phone.
Yeah.
So your car, you see that car over there? I'll take that.
But we could do biology things too.
Yeah. My biggest frustration in my own life as a research scientist was to go down to the Department of Agriculture and try to interface with the people interested in policy for whether or not we should online screen for parasites.
Mm.
And that was a difficult sell. In fact, I didn't sell it, and to the frustration of the scientists that worked at USDA as well. So to get people to realize what the impact of those policies are and why you're actually working on these things to begin with, they require open minds, and sometimes the minds are not as well open as you'd like them to be.
Let's do 2 more emails.
Okay.
And,
before you do that, I wanted to comment on the fact that the article by the second 2 authors, Diane de Ulis and Gigi Gronval, refer to this thing called the Imperiale report. And I was unfamiliar with that report itself, even though Mike Imperiale is just downstairs for me. So I tracked that down. And they are both part of the members of the committee that put this report together. And Mike was the chair of the committee. And then interestingly, they list the people who reviewed the report as well. The report is downloadable for free from the National Academy's site. And so if you go to the open access M-Sphere article, the second one, and you can find the reference there. It's reference 6 if you want to look at that. So the report itself is, or the committee is Committee on Strategies for Identifying and Addressing Biodefense Vulnerabilities Posed by Synthetic Biology. So, and it's an interim report. So
wonderful. Dickson, can you take the next email?
Carrie writes, "Hello, TWiV team. I adore your podcast and the related podcasts, TWiP, TWiVo, and TWIM. I can't emphasize enough how helpful it is to have current research and scientific events put into perspective by such knowledgeable experts. As a graduate student in a disease ecology lab, I find these podcasts an invaluable resource. I wish I had discovered them sooner, but at least it will be a very long time before I run out of quality educational entertainment. Keep up the great work."
Thank you, Carrie. Kathy.
Ken writes, "Dear TWiV podcasters, I recently read that our layer of dead skin protects against viruses since they can't interact with dead cells. So is there evidence that people that actively remove dead skin by exfoliation, for example with pumice stones, are more susceptible to viral infections?" Best, Ken, and he's from Oxford. And so Vincent has made a comment and inserted a diagram. So go ahead. Take it away, Vincent.
I have a diagram of the skin, and the upper layers that are dead, they're called the stratum corneum. It's 15 to 20 layers of dead cells. And then under those are the living cells. So I don't think you could remove all those layers from enough parts of your body to make yourself susceptible. And then, of course, they grow back. The dead skin is replaced as the cells below it continue to die. So, you'd have to exfoliate your entire body, all these 20 layers. So I don't think that it's going to increase. And I don't know of any studies that address this, but, locally, on the girl's face, she scratched her face. And that's a scratch. So that's the thing.
That's a good point.
That got down below that layer, I'm sure.
If you scratch it, you could. But so exfoliation could scratch you. And so, yes, if you exfoliated, let's say, your hand and then went and petted your cat and the cat had cowpox on it, then, yeah, you're right.
Yeah, if you exfoliated to the point of tissue damage, I mean, where it hurt and you were rubbing your skin raw and perhaps even to bleeding, then it could be an issue.
So yes, in light of our snippet today, it could. It takes a little scratch, and a exfoliation could scratch you. So you could. Yeah, I was interpreting it as removing it all permanently, and then you could walk around for weeks and be susceptible. But yeah, it could be local things. So. Yeah.
So why do I want to exfoliate anyway?
That is another podcast.
It's to get rid of your wrinkles, Rich.
I don't know.
They can't age by looking at them.
I'm okay with the way things are. Yeah.
All right, let's do some picks. Rich, Condit, what do you have for us?
Okay, I have one of my typically complex picks. The primary pick is the Brighton collaboration. Which I'm really surprised I have not picked before. And a sub-pick within the Brighton collaboration is a thing called the Viral Vector Vaccine Safety Working Group. The link to the Brighton collaboration links to the Brighton collaboration site. The Viral Vaccine Safety Working Group links to a paper that describes that group. And then there's another link that I'll get to in a minute. The Brighton collaboration is an organization that is dedicated to improving vaccine safety on an international scale and putting together an international collaboration to share information and to standardize things like case descriptions of adverse effects and promote communication about adverse effects, and also promote communication and standardization of vaccine formulations, et cetera. It is the brainchild of a scientist named Robert Chen at the CDC. And that's where the third link comes from, to remind you of a pick that we made some time ago, an article by Chen and DiStefano called Vaccine Adverse Effects, Causal or Coincidental, which is the commentary that was in The Lancet at the same time as the Wakefield article. That said that the Wakefield article is seriously flawed and, if taken seriously, could do damage. And it was absolutely prescient. So Bob Chen, the founder of the and really force behind the Brighton collaboration, in my opinion, has serious cred as the author of that article, and he has many other accomplishments as well. The reason I bring this up now is that I've been volunteering for this organization for, God, I don't know, I think at least 8 years, and I've just recently stepped down. And I'm not going to do that anymore. And I was on the viral vector vaccine safety working group, which is described in the other link, which actually was quite interesting. We met monthly in an international conference call, and the mission of the working group, the primary mission, is to develop a template that describes different viral vectors for vaccines that people can understand, so that if a vectored vaccine comes up and you want to know what it consists of, you can look at a standardized template that describes this. As you might imagine, a committee that meets by phone once a month for an hour does not make real rapid progress on something like this, but it's plugging ahead. And, the people on the committee consist of regulators and scientists and biotech people, a fascinating collection of people and a collection of perspectives who are really dedicated to pushing this whole thing forward. So it was a very informative experience for me and a rewarding experience. I enjoyed it, but it's time for me to go. And one of the reasons I'm bringing this up now is that they're always looking for other people. So if this wrap interests you and you are a scientist who you think could contribute to this effort and would be interesting, it's not too time consuming and it's very interesting, either contact Bob Chen of the Brighton collaboration directly through these links, or contact me and I'll put you in touch with him. All
right. Here you go, listeners. Step forward. Dickson, what do you have for us?
I have a pick for Rich. That is the amazing details of Jupiter's clouds by the Juno mission, and it's fabulous stuff. I mean, it's just it is unbelievable.
And that's a painting.
I know. It's a painting. That's right. Nature's paintings, and
now he's got a painting. It's a Van Gogh.
That's right. There's tons of Starry Night. Exactly right,
beautiful,
Rich.
Yeah.
And if you go to the website of NASA and then hit Juno mission, you can see many more of these, and they're just astounding.
Make a great desktop.
The reason why there aren't more pictures from this mission is because of the size of the orbit of this satellite. Mm-hmm. It has to go way outside of Jupiter to come back down again, so that it avoids all of the magnetic fields.
How long does that take?
It takes about a week and a half or 2 weeks, something like this, for a complete orbit. So they don't get many pictures, because imagine how fast this thing is going when it's only about 10,000 miles away from the surface. I mean, it's like, how does it even get a picture in focus, for gosh sakes? Because there's no time to take it, right? But yet it does it anyway. So it's a fabulous mission, and it's got about another year, I think they said, and then that's the end. My second pick is an unusual pick for me, because obviously I'm picking a virus article, and I don't usually do that. But I was fascinated by the Epstein-Barr transcription factor Na2 as a homologue to a variety of sites in the mammalian genome which results in autoimmune diseases. And one of the big ones is, of course, systemic lupus erythematosus. But there are others listed here. There are about 7 different autoimmune diseases that this transcription factor actually fit onto that would be a possible cause of that autoimmune disease. And I was absolutely stunned to see. And
the idea is that EBNA2 protein would stimulate an immune response that would have cross-reactivity with yourself, so stimulate.
Sure.
And so this is Epstein-Barr virus, which is ubiquitous.
Right.
That's very interesting.
Yeah. And so when Vincent asked the question at the Science March, how many of you think you have a virus infection? 2 hands went up. Oh, and I meant to ask it differently. How many people here have ever had infectious mononucleosis? Everybody's hand goes up, right?
Yeah. I wanted to know at the moment.
Yeah, I know, right. Well, I was knocked out by the amount of work that this thing entailed. This was about an 8-year project, and they did it by computer, basically. They did it by a series of programs. They even had to write 2 new ones in order to get this thing to actually work. And now that they've got it working, this was done in Cincinnati, by the way, at the University of Cincinnati at the medical school. They're going to release this program to everybody so they can take similar transcription factors from other viral infections and see whether they match up with things that equal disease in humans.
So what I thought interesting about this, so the name of the paper is Transcription Factors Operate Across Disease Loci with EBNA2 Implicated in Autoimmunity. This idea that they use computational methods, and you can see that transcription factors can work at loci that are involved in different genetic disorders, right?
Yes.
So sometimes you try and do these associations of mutations with disease, and it doesn't make sense. But here, a factor is what's going on.
7
and somehow
places, same
It's very
effect.
cool.
It's very cool. So I like articles like that. You're welcome.
Kathy, what do you have?
I have a picture, Dickson, sort of. It's one that my friend Isabel sent me, and it's also something for all the Hispanic speaking listeners that we have, because all of the text is in Spanish. But the overall headline is that these are landscapes that look like they're from another world, but they're really just from Spain. And some of them are quite remarkable. And so I just thought it was a set of really neat-looking pictures that
Fabulous.
Amazing.
Could check
Fabulous.
out.
It's amazing.
Fabulous.
Oh, my gosh.
Thank you for the eye candy.
It's really nice. Yeah, it's just some of these are stunning. Wow.
This one looks like a Greek amphitheater.
Yeah.
Look at that
But
green.
it's not a Greek amphitheater.
Dixon, look at that green lake.
Right. Well,
Got algae in it.
I'm not there yet. I'm going to scroll down.
Kind of otherworldly green.
Charco de los Clicos in Lanzarote.
Wow.
Nice. Very nice.
Cool.
Yeah, very cool. Spain has a wide variety of biomes, I must say.
It does. Mm-hmm. I've been there.
It's a big place.
Spain. It is.
My pick is not as uplifting as all the others. Mine is a article in Nature Biotechnology, Evidence for a Mental Health Crisis in Graduate Education. This is an article about a study that was done of 2,279 individuals, 90% PhD and 10% master's students from 26 countries, 234 institutions, a diverse variety of fields, not just biology. And they gave them questionnaires, and they show that graduate students are more than 6 times as likely to experience depression and anxiety compared to the general population. And they have graphs showing 41% had anxiety, 39% depression. And they try and explore some of the reasons. If you're male, 34% had anxiety. If you're female, 43% had anxiety. If you're transgender, 55% had anxiety. So being transgender in this field is obviously not only leading to anxiety, but depression. And then they try, there's one where a lot of them say their mentor makes them anxious or
tormentor.
So it's unfortunate because, as we've said on Twitter, there are a lot of problems with science, and it's kind of amplified here in this study. And the PI is part of it too.
But doing research is frustrating.
Yeah, but it's also, that's always been the case. But now there are other things that are making it harder, right?
If
like
you're a student with a
and
time frame in which to finish
job,
and your experiments are not going well, that leads to anxiety, I think.
I think this probably, if this had been done 30 years ago, would have been very different. No, I don't think it would be the same.
So?
No, I think
I think 'twas ever thus. If your
'twas
stuff
ever thus?
wasn't working, yeah.
You would be depressed and anxious.
I do too.
I don't know. I think it's transient though, because then if you hit someone when everything is fine. But I think there are so many other things. I mean, you could be right, but I think there's so many other things now that are worse than 40 years ago that maybe that contributes to it. Anyway, I don't know if it's open access, but it's interesting. It's not.
And I don't think you mean transient necessarily. I mean, because if it's a physiological depression that's, compounded by this, then it's not necessarily transient.
No, but you may ask someone, are you depressed? And they say, yeah, this week none of my experiments are working, so I'm depressed. And then maybe if you hit them and when things are working or when they're about to graduate, you would get a different answer. But I agree that true depression is not transient, but the way they answer the survey, it may be, depending on the survey. I don't know. All right, that is TWiV 490. We're at microbe.tv/TWiV. Send us your questions and comments to twiv@microbe.tv. And if you do like what we do, please support us financially. Go to microbe.tv, contribute. We have multiple ways you can do that. Dickson Despommier can be found at parasiteswithoutborders.com.
The Living River.
thelivingriver.org.
We are reviving
Racaniello.org.
Yep.
Yeah, we're working on it.
It's going to be back.
Thank you, Dickson.
You're welcome.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. We just got a text from Alan Dove, who is in, Washington, D.C.?
Oh.
Yeah, it's a picture of the right glider.
That's right.
Oh, nice.
So would that be the Air and Space Museum?
Yeah.
No, the Smithsonian thingy, right?
Smithsonian?
Right.
Yeah, down at the mall there.
Same thing.
But that's part of it. The Aerospace Museum is the Smithsonian.
Part of the Smithsonian.
It is.
Yeah. Thanks ASM for their support of TWiV and Ronald Jenkins for his music, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.